Good morning. I will talk today about. I will start with the starting material of steroid hormone synthesis, cholesterol, its sources and transportation in the blood to target tissues. Then I will talk about the sites of steroid synthesis, the specific enzymes, and the molecular basis of lack of certain enzymes in the steroid synthetic pathway, which results in different congenital adrenal hyperplasia. The starting material for steroid hormone synthesis is cholesterol. Cholesterol can come from dietary sources. We eat a lot of cholesterol-rich food, and cholesterol is taken up by the digestive system and packaged into chylomicrons. Chylomicrons are lipoproteins that are assembled to deliver lipid-based molecules to the target cells. Chylomicrons go to the circulation, release fats, release lipids, and the remnants are coming back to the liver, so the excess cholesterol that we have taken up comes to the liver for elimination. Liver can also synthesize de novo cholesterol. The two, the dietary cholesterol and the de novo synthesized cholesterol, need to be in good balance so that we don't have too much cholesterol circulating in our blood. Some extrahepatic tissues can synthesize cholesterol, but usually they don't release it, they use it for further processing. So let's remember that the de novo synthesis is mainly happening in the liver, and liver is also taking care of eliminating excess cholesterol coming from chylomicron remnants or coming from the periphery delivered by HDL or high density lipoprotein. Cholesterol is synthesized, released in the form of VLDL, coming back in the form of HDL and chylomicron remnants. Thus, liver needs to do something with this excess cholesterol. Sometimes it is repackaged and goes back to the circulation, but also the excess cholesterol needs to be eliminated, and it mainly is going with conversion the cholesterol to bile solubilizing cholesterol and eliminates it down to the small intestine. Just a note that steroid hormones that are synthesized from cholesterol are also eliminated by the liver. Liver being the main detoxification center of our body, it's important to have enzymes and systems that handle the excess of steroid hormones too. Cholesterol is a very important molecule. It is sitting in every membrane of ours, let it be plasma membrane or intracellular membrane of organelles. Where we have more cholesterol, the membrane is more rigid. We can turn it into bile acids, bile salts that are very important in digesting lipid in the small intestine from food. Precursor of steroid hormones, which is the topic of today's lecture. Progesterone, glucocorticoids, mineralocorticoids, and sex hormones are synthesized from cholesterol. We can also produce vitamin D precursors and derivatives, and cholesterol is helping. Cholesterol is synthesized mainly in the liver, as I mentioned. However, adrenal cortex and reproductive organs, ovaries, testes, placenta, may be able to synthesize some, but these cells can also take up cholesterol from the circulation to produce steroid hormones in situ. Cholesterol source is acetyl coenzyme A, which comes mainly from glucose and synthesized in the liver, as I mentioned. It's a very expensive process. ATP and NADPH from the hexose monophosphate shunt is important. So everything which cholesterol is synthesized from in the liver comes from glucose. cytochrome P450 system is a heme protein, an enzyme system localized either in the mitochondria or smooth endoplasmic reticulum of a couple of organs. It uses molecular oxygen and NADPH, again comes from the hexose monophosphate shunt, and catalyzes oxidation reaction. We call it monooxygenase, which means that it uses one atom of the molecular oxygen plus NADPH to modify target molecules. We use it vastly in the SIP system. All mammalian cells except for mature red blood cells and skeletal muscle uses the cytochrome P450 enzyme system for something. What do we use cytochrome P450? Of course, steroid hormone synthesis, this is what we talk about today, but certain metabolic processes in fatty acids, prostaglandins, leukotrienes, retinoids, vitamin D, all use a certain class or a certain type of cytochrome P50. Activation, inactivation of therapeutic agents is important, as well as detoxification reactions, mainly in the liver, to eliminate chemicals, to drugs, to insoluble byproducts of metabolism. So this family, the SIP family or cytochrome P450 family, is a very widespread enzyme system. We have a lot of genes that code for a SIP. So how do we distinguish from one SIP from another? SIP stands for the protein or the gene of the cytochrome P450 family. Which specific family within the cytochrome P450 big family we are talking about is labeled by the Arabic number. A subfamily with an additional capital letter, and the individual P450 in that family is labeled with an addition of another Arabic number. These are the SIPs that I will talk about here in this lecture. CYP11A1, CYP11B1, CYP11B2. You see these are members of the CYP11 family, but one is the subfamily A, the other is subfamily B, and within subfamily B, we actually utilize two members, B1 and B2. CYP17, CYP19 will also be mentioned. You see that some of these enzymes have specific names. CYP11A1, a concrete enzyme, which I will talk about in a second, is called desmolase. This is the rate-limiting committed step of steroid hormone synthesis. And CYP19 is aromatase. In between, we have CYP11, which is 11-beta hydroxylase. The other member, the B2, is 18-alpha hydroxylase, and CYP17 is 17-alpha hydroxylase. 11, 18, or 19 actually, and 17 means which carbon atom of the cholesterol ring is getting a modification by the SIP system, either hydroxylation or further oxidation. Alright, so this is actually the same figure. Unfortunately, it is flip-flopped, but bear with me. So we start with cholesterol. Cholesterol is a 27 carbon number complex molecule having a very strict ring system and an aliphatic chain. The first SIP, the CYP11A1 or desmolase, turns cholesterol to pregnenolone. From pregnenolone, we are going to progesterone, and progesterone we are synthesizing the glucocorticoids, mineralocorticoids, and sex hormones. Here, glucocorticoids, mineralocorticoids, or sex hormones. The glucocorticoids and mineralocorticoids together we call corticosteroids because the synthesis of these two classes of hormones is happening in the adrenal cortex. Glucocorticoids like cortisol, mineralocorticoids like aldosterone, all synthesized in the adrenal cortex cells. Sex hormones in the ovaries or placenta like estrogen, estradiol, and the testosterone or the active form dihydrotestosterone also synthesized in the testes. Now, this enzyme, the P450 monooxygenases, can sit either in the mitochondria or in the smooth endoplasmic reticulum. So I will talk about this sublocal in a second. Let's just have 1 second spent on CYP11A1 or desmolase. This is actually catalyzing the turn of cholesterol to pregnenolone, but actually these are 3 independent enzymatic steps catalyzed by the same enzyme. See, all of these use NADPH and molecular oxygen, and what we end up with is nothing else like turning this longer aliphatic side chain of cholesterol to a shorter one. So C27 goes to C21. And then we go further and modify pregnenolone to get the different classes and different types of steroids. How do we do that? This, what is depicted in this particular figure, is happening in the adrenal cortex. Different zones. The blue depicts aldosterone or mineralocorticoid synthesis, and these enzymatic steps are in the zona glomerulosa. MIT means mitochondrial enzyme, ER means smooth endoplasmic reticulum enzyme. Glucocorticoids, labeled by yellow circle, are synthesized in the zona fasciculata, and the androgens, like testosterone, can be synthesized in the zona reticularis. These hormones then released, carried in the blood by specific carrier proteins, and delivered to the target cells. So let's focus just for a second. So this is what we talked about. This is CYP11A1 in the mitochondrium, the cells in the zona glomerulosa that produce pregnenolone. And from pregnenolone, we go further to cortisols and even further to testosterone synthesis, to androgen synthesis. Pregnenolone then can go down, and with the involvement of CYP21, the 21 hydroxylase, the CYP11 hydroxylase, CYP11B2, the 18 hydroxylase, we end up with aldosterone. Now CYP17, the 17 hydroxylases, work in the ER of the zona fasciculata cells, and with the CYP21A2, same enzyme, which were acting in the ER in the zona glomerulosa, will act in the cells in the zona fasciculata and produces deoxycortisol and cortisol with the CYP11 in the mitochondrium. Now these two 17 hydroxylated byproducts from pregnenolone and progesterone can be further modified in the zona reticularis in the ER to go down in androgen synthesis. Please note, however, that the active androgens, dihydrotestosterone, synthesized in the testes, and estrogens from androgens are synthesized in the ovaries and the pregnant placenta by aromatase or CYP19 enzymes. Why do you need to know about these enzyme systems? Because unfortunately, we know genetic disorders where one or more enzymes of the steroid hormone synthetic pathways are not produced missing because of point mutation or other mutations. So let me go down in this pathway. You see steroid hormone synthetic pathway that we went through until now, starting from cholesterol, desmolase, pregnenolone, going to progesterone. Progesterone is the junction point of between aldosterone or the other steroid hormones, and then an additional step will go for glucocorticoid or androgen and later estrogen production. So what if one or the other enzyme is missing from this complex pathway? The problem is very serious. Luckily, this is a very rare disorder. If the very first enzyme after pregnenolone, the 3-beta hydroxysteroid dehydrogenase, is not produced. Why is it the most serious? Because it halts all steroid hormone synthesis. There is no aldosterone, no cortisol, and no androgen or estrogen synthesis can go on. Luckily, as I said, it's very rare. 17-hydroxylase deficiency will result in no production of sex hormones and no production of glucocorticoids. So what will happen in this case? There is no problem with mineralocorticoid synthesis. It will be synthesized. But because it is increased, because the system feels that more steroid hormone needs to be synthesized, but it can go only towards mineralocorticoids, there will be a big problem with sodium and fluid retention, hypertension, and whatsoever. And of course, the sexual development and some metabolic problems will occur. I put here for you on the side the 21-alpha hydroxylase deficiency or CYP21 deficiency, because that is the most common. Actually, about 90% of the known congenital adrenal hyperplasia patients are missing this particular enzyme. What is the consequence of that? 21-hydroxylase acts on two pathways. One is the synthesis of mineralocorticoids and the other is the synthesis of cortisol. So the consequence is that there will be no glucocorticoid and no mineralocorticoid synthesis. However, the push will go towards androgen synthesis. So there will be a problem with blood pressure, there will be a problem with mineral and fluid balance, there will be a partial problem with metabolism, and there will be a problem because of overproduction of androgens, early virilization of the males, and precocious sex organ development in female babies. So the last one that I wanted to mention is the 11-beta hydroxylase deficiency. Again, it is rare. CYP11B1 and B2 deficiency. This is majorly B1. The problem is that aldosterone and cortisol will not be synthesized. Precursors will be there, but again, the problem will be that a lot of androgens will be synthesized, so vascularization, virilization also is a sign of 11-beta hydroxylase deficiency or CYP11B1 deficiency. So what did we learn today? That steroid hormones starting material is cholesterol. Cholesterol is coming from either food or newly synthesized, de novo synthesized. Elimination of the excess cholesterol and steroid hormones, as well as the majority of de novo synthesis of cholesterol, happens in the liver. However, steroid hormones are not synthesized in the liver. They are synthesized in target organs, mainly the adrenal cortex and some intestines, ovaries and placenta, androgens and estrogens. Many of the enzymes belong to the cytochrome P450 system, These are monooxygenases working with molecular oxygen and NADPH. And depending on their localization, either in the adrenal cortex, mitochondria, or smooth endoplasmic reticulum, we can synthesize corticosteroids, mineral corticoids, glucocorticoids, and some androgens in the adrenal cortex, while the active androgens are synthesized in testes and estrogens are synthesized in the ovaries or in the placenta. The committed step of the whole synthetic process is so-called desmolase. That also is a CYP enzyme, and the further catalysis is done, catalytic steps are done with other CYPs, CYP11 B family members, CYP17, 18, or 19. To remember that either of these enzymes are missing because of genetic disorders, congenital adrenal hyperplasias will develop, and these are due to CYP deficiencies. They are influencing our balance, or actually producing imbalance in steroid hormones in the body. Thank you for your attention.